At a molecular level, raw genomic data is dense, abstract, and heavily encoded. When researchers sequence a new strand of DNA, they extract a string of millions of A, T, C, and G nucleotides. On its own, this isolated sequence is entirely enigmatic. To determine its origin or its biological function, that sequence must be cross-referenced against biological entities we already understand. The obstacle is scale. Global genomic databases are expanding at an exponential rate, capturing a volume of biological data that is difficult to process. Taking one mystery sequence and manually comparing it against the millions of documented sequences currently stored on global servers is a mathematical impossibility. Traditional alignment methods struggle to scale to this size. When you force standard alignment computations against datasets this massive, the system creates a bottleneck that stalls biological research. Without a high-speed computational solution to compare and align these strands, modern genomic sequencing generates noise devoid of scientific value. The standardized computational solution to this alignment bottleneck is NCBI BLAST, the basic local alignment search tool. Rather than relying on slow, exhaustive alignment calculations, BLAST operates as a high-speed heuristic filter. It quickly estimates alignments, rapidly comparing an unknown query sequence against vast libraries of known genomes to find regions of local similarity. Using BLAST effectively requires more than executing a search. It demands precise algorithmic selection, strategic targeting of the database, rigorous statistical analysis of the output. Mastering this computational workflow provides the foundational framework for transforming raw DNA from an abstract text file into actionable evolutionary or medical discoveries. Sequence searching is not a one-size-fits-all process. The first step requires researchers to select the correct algorithmic pathway based entirely on their specific input data and biological goal. BLAST-N is engineered for nucleotide sequences, allowing researchers to compare a raw strand of DNA or RNA directly against a nucleotide database. Conversely, BLAST-P is built for protein researchers, comparing an amino acid input directly against a database of known proteins. The math becomes far more complex when researchers need to bridge the gap between raw DNA and functional proteins. That requires BLAST-X. BLAST-X mathematically translates a raw, uncharacterized nucleotide sequence into its corresponding amino acids before it executes the search against the protein database. This specific cross-translation is essential when researchers are hunting for potential gene products hidden within large stretches of unannotated DNA. Algorithmic specificity is the critical first step. Choosing an incorrect program mathematically guarantees an inaccurate biological conclusion. Once the algorithmic pathway is set, researchers must select the target database to query against. It is often assumed that selecting the largest, most comprehensive global database will yield the most comprehensive result. In reality, querying an unfocused database introduces overwhelming computational noise. It forces the algorithm to return distant, irrelevant evolutionary matches that bury the actual biological signal. The solution involves narrowing down the search to highly focused, expertly curated databases. NCBI's Core NT is a specialized database built to provide clean, reliable data. Targeting a focused database drastically reduces unnecessary compute time while simultaneously increasing the relevance of the biological match. The software side has also evolved to manage these workloads. The BLAST Plus suite utilizes multithreading, allowing local computers to process searches across multiple CPU cores simultaneously. For modern genomics, massive workloads are now offloaded to cloud-based solutions like Elastic BLAST, scaling computational power dynamically to meet the growing volume of sequence data.
But algorithmic power is useless without precision. Targeting the correct database is the only way to separate biological signal from computational noise. Upon completion, a BLAST search generates a complex matrix of statistics requiring careful interpretation. This graphic represents a standard BLAST output dashboard. To determine if an alignment is a true biological match, researchers must evaluate 4 specific columns simultaneously: query coverage, percentage identity, E-value, and bit score. The baseline alignment process scores the literal physical match between your query and the database sequence, rigorously accounting for genetic mismatches and physical gaps. This generates the bit score. This metric represents the absolute mathematical strength of the match, factoring in the length of the alignment against the penalties incurred by any gaps. The critical statistical anchor of the entire system is the expect value, or E-value. The E-value measures the statistical likelihood that this specific sequence alignment occurred entirely by random chance within a database of this precise size. This metric operates on an inverse scale. Lower numbers mean higher confidence. An E-value approaching 0.0 represents virtually zero probability of a random coincidental match. However, a low E-value and a high bit score mean nothing in a vacuum. Isolating these numbers without contextualizing the physical coverage of the sequence is dangerous. True genetic homology is a synthesized statistical probability. Reading the entire dashboard holistically is mandatory. The most common and dangerous mistake made by early career bioinformatics researchers is treating the top hit on the dashboard as the absolute ground truth. Consider a scenario where a researcher receives a search result boasting a 100% percentage identity. The fatal flaw in this scenario is revealed by checking the query coverage. That 100% match only covers a tiny 5% fraction of the total sequence length. This constitutes a biological false positive. The small matched segment may be a common functional domain shared by many organisms, but the overall sequences belong to entirely distinct biological entities. Relying solely on percentage identity without verifying the total query coverage leads to flawed genomic discoveries and corrupted downstream research. Algorithmic outputs require manual verification. Human biological intuition is necessary to review the math and determine if the physical match makes sense. Statistics can be mathematically accurate, but biologically false. Synthesizing coverage, identity, and the E-value is the only way to prove a sequence match is reality. The holistic BLAST workflow is a rigorous pipeline, inputting the data, selecting the specific program, targeting a curated database, and validating the output statistics. This exact workflow is executed daily for gene annotation. It allows scientists to predict the function and physical location of genes within newly mapped genomes. BLAST outputs also allow researchers to trace evolutionary lineages, quantifying the biological similarities across organisms to map millions of years of genetic divergence. In time-sensitive clinical environments, researchers rely on BLAST to sequence and rapidly identify unknown, aggressively multiplying pathogens before they spread. These rapid identifications directly support biotechnology applications, accelerating targeted drug discovery and providing the genetic data required for personalized medicine. Artificial intelligence and cloud computing will increasingly automate these pipelines, speeding up the time between a query and a result. But the foundational logic of heuristic comparison will remain. As long as uncharacterized data hides within DNA, NCBI BLAST remains the essential computational bridge between raw sequences and meaningful scientific discovery.